Yes, welcome to the day 11 session on T cell epitope prediction in internship and certificate course in artificial intelligence and machine learning approaches of drug and vaccine design. So now before going to today's session topics, can we recap the last session topics? What are the last session topics we have discussed? You want me to show some hints to see what exactly did we discuss in the last session? Yes, we initiated with immunoinformatics. Immunoinformatics, also called as computational immunology, isn't it? That's what we have discussed. And what else did we discuss? Only one word. How about the others? So, what else did we discuss other than the immunoinformatics? Vaccinology, very good. We distinguish vaccinology and reverse vaccinology. That is conventional vaccinology and reverse vaccinology. And then we have discussed the examples for the types of vaccines. And then what else did we discuss? Did we discuss types of epitopes? What are the types of epitopes? What are the major classes of epitopes? Yeah, participants, come with your answers. Come fast. SVC students, are you available? How about others? Yes, very good. Linear and continuous. Linear and continuous is for B cell epitope. Very good. So this is for B cell. So see, if only if I ask you to ask the answer, you will be answering. You can answer, okay, if you know the answers. Very good. So we have two types of epitopes, T cell epitope and B cell epitope. Good. So you're listening to my class. Now let me go ahead with today's session topics because still we have lot of contents to see. So today I'll be taking you directly to the website called iedb.org. So IEDB also called as Immune Epitope Database. Okay, before that, I can also ask few questions. Whatever questions I'm asking, these questions you can find in MCQ, final MCQ. So only I'm raising these kind of a questions, and even this could kind of an interaction will motivate others also. The next question is, which is the first vaccine discovered from reverse vaccinology? Discovered from reverse vaccinology. Hmm. Vaccinology. Spelling wrong. Very good. The answer is meningococcus B polysaccharide vaccine. Meningococcus B polysaccharide vaccine is used for treatment of is used for okay sorry is used for the treatment of bacterial meningitis. I like to remove this 1 minute. So the text is coming. Do you know how to stop this text? Yes, now it's okay. I share once again. So ietb.org, that is immune epitope database. In immune epitope database, we have types of epitopes like T cell epitope, B cell epitope. In T cell epitope, you remember we have two types of epitope, that is helper T cell epitope and cytotoxic T cell epitope. And so if you want to predict T cell epitope prediction, we have numerous tools inside. Okay, so if I click this link, you can able to see the following programs. So whatever I clicked, initially is T cell epitope prediction for class 1 MHC molecule. Similarly, you can go with, if you want to predict MSC class 2 epitope, you can click this. Okay. And we also have epitope analysis tools. Today we'll be discussing on T cell epitope prediction tools and then epitope analysis tools, how to use this. And you can see this is the latest data updated in these databases. Peptide epitopes you can find around 17 lakh structures, 17.4 lakh structures. T cell assays, B cell assays, whatever done in wet lab reported from this much amount of peptides or deposited. Not only we can see the epitope peptides for T cell and B cell, but if you want to search for even peptides for any specific disease, for example, asthma. Asthma is an autoimmune disease. So if you click this find, you'll be getting, it asks you to type any kind of a disease. If you click for asthma and you search it, you can see types of asthma, allergic asthma, chronic asthma. So if you want to get inside, you can select this, okay, and we can able to find the information for this. Even if you want to find specifically here, like disease, autoimmune disease, and then it is related to asthma is pulmonary. Pulmonary is here, not here. Somewhere is missing. It can be endocrine, exocrine. Okay. If I take some other kind of autoimmune disease, for example, atherosclerosis. Okay, I'll be selecting the atherosclerosis and I have to click apply. Or if I want to go with the already selected asthma, you can search for the asthma, you can select here. And if you click apply, okay, allergic asthma and atherosclerosis, both are selected at present. So you're getting two. Now you have to click search. So for whatever diseases we have selected, we'll be getting the peptides. Okay. So these are the peptide sequences, epitope peptide sequences. Okay, and you can also see the references and you can see the organism. So not only we can see for Homo sapiens, but we can also see for the other animals, especially the pet animals. Other than this, we can also see something like camel. So I can show you what else we can find the data. Now you find this, whatever I searched for the two different diseases, the epitopic peptides used for the treatment for of various organisms are shown here. So around 1,200 data are available. Okay. So you can find which is you can try to screen even the peptides for one particular organism, and then whatever you screened as the best peptide, that you can even check in docking and simulation, and we can finally validate that peptide can be used for effective treatment of that specific disease or not. Okay, so this is how we can search the peptides with respect to certain diseases, and also with respect to certain organisms we can use this host. Okay, so similarly if you click find, okay, you can find bird, aves, fish, mammal. So if you want to focus specifically the horse, equus caballus, as I'm selecting this apply. Okay, and if you click search, so whatever we selected for a particular mammal, so we'll be getting the corresponding epitopes. Yeah, meningococcal. So any doubts you can ask me. Are you asking any doubts? Do you have any doubts? If no, I just go ahead. I close the WhatsApp. Now let me go ahead with so you'll be getting these epitopes here. We can also get antigens, assays, receptors, references, etc. Other than whatever epitopes we'll be getting here. So one thing you can remember that we can able to search for T cell and B cell epitopes that from the already existing databases present in IEDB. IEDB stands for Immune Epitope Database and Tools. Okay, but if I want to specifically search epitopes for Nipah virus or for SARS-CoV-2 virus, then we can able to give the particular protein, whatever protein. See here also, if you see for the horse, we'll can able to get the description of antigen like GAG, polyprotein, envelope glycoprotein, large envelope protein. So like this, we have different types of antigens and specific organisms. So what virus? Like if the horse is infected from hepatitis B virus, so what are the different protein targets available? One such target is large envelope protein. And for this, this is the best peptide epitope already deposited. So like this, we can also have few more peptides also for the same virus, same protein. So, but whatever we get this data, we can take it and we can screen. Okay, there are so many properties in the peptides to screen. We can screen here and we can also go screen the other filters like using a docking simulation, etc. Okay, now let me go ahead with the today's session content on T cell epitope prediction. Just I started with the EDB. What else we can practice with this tools of IEDB? Let me go ahead. So today we'll be discussing on workflow, like you use a sequence of steps to work on a particular research, that is especially searching for T cell epitope. And then we'll be discussing on the role, mechanism, genes, and function of the MSC molecules. Of course, we have discussed on yesterday. Just a recap, a difference between MSC molecules, immunological database, and what are the other databases we have for T cell epitope prediction. That also we'll be discussing. And other than that, we'll be discussing something called immunogenicity, conservation, okay, conservation, toxicity analysis, and COVID-19 vaccine adjuvants. So now T cell epitope prediction, as I earlier mentioned, whatever we see at the top is the helper T cell is going to interact with what molecule? MSC2. So initially we try with the MSC1 prediction, isn't it? Whatever MSC1 prediction we find in IEDB is for the cytotoxic T cells. Okay, and related to the shape of the T cell and B cell receptor. So in T cell epitope, we are concerning about the T cell receptor. You know, the T cell receptor, whatever we can see here, this T cell receptor alpha beta is present here, something like this. And these are surrounded by CD3 receptors. The CD3 receptors are present in quadruple. So this is how the CD3 receptors are surrounded surrounded by the T cell receptor. So this is a side view. In the top view, you can find like this. Okay, and this is what interacting with the MHC molecule of APC. And whatever I mentioned earlier, okay, a large diagram, large figure I am showing it to you. One thing you have to remember, whatever I am showing you as a helper T cell, okay, is going to have, that is MHC, MHC2. MHC2, whatever I am showing, is going to have a larger size of peptide when compared to the cytotoxic T cell. So whatever initially we predict MHC1, we predict around 9 amino acid peptide. And for this, we predict around 13 amino acid. I told you the reason. The reason behind is where exactly our peptide is presenting. If you see in MHC molecule 1, yeah, this is one subunit. Alpha 1 and alpha 2 is one macroglobulin subunit. Macroglobulins subunit. So just because of this, we need just a small size, but whereas this is a two different subunits, so we have a large size of a peptide, so around 13 amino acids. This I already explained you what is most important for T cell activation, not just CD4 or CD8 along with the T cell receptor interacting with MSC2 or 1 can activate. You must have this kind of an interaction also, CD80 and CD28. If and if the CD80 or CD86 of dendritic cell or any antigen presenting cell interacts with CD28 of T cell, then only T cell gets activated along with this. So 1 and 2, these two interactions are equally important to get T cell activated. If one is failing, the activation won't happen. What is the purpose of activation? If and if the T cell activated, then only we can see the T cell is going to proliferate into two T cells. Other than this, it can also release some kind of a cytokines. These cytokines also can activate APCs, or sometimes even it can activate T cell also. Now related to roles of MSC molecules. This whatever I'm showing is the MSC1 antigen presenting processing and presenting pathway. I told you if a virus or bacteria entering inside degraded by the proteasome and the transport associated protein can transport only the peptides, the cleaved peptides, and this will be presented on the MSC molecule, which is transported through Golgi apparatus, finally transported through secretory vesicle, and the final presentation after the processing, you can able to see that antigen is presenting on the MSC molecule, which is present in the surface receptor of the APC, which is present as surface receptor of APC. And if this interact with the T cell epitope, okay, so whatever binding region in the T cell, this is what we call it as epitope. Okay, and so this T cell epitope only we are predicting. Now let me go ahead with this structure of MHC molecules. Already we have discussed the single macroglobulin, multiple macroglobulin. The one thing we should remember, we should not forget is the class 1 MHC molecules or HLA, HLA, A, comma, B, comma, C. What is the class 1 MHC is generally a name of a protein and the corresponding genes are HLA, that is human leukocyte antigen. Okay, class 1 MHC protein, I mean HLA genes code for class 1 MHC proteins, and these are the main genes. And similarly for class MHC2, HLA, T, P, D, Q, D, R. These are the three important genes we have to remember. Difference between major histocompatibility complex molecules we can see in multiple immunology textbooks. Okay, any textbooks you can find this information, because I told you whatever HLA genes or MHC molecules that distinguishes from individual to individual. Even siblings have distinguished HLA genes. Okay, only the HLA genes can infer whether you can able to treat with any particular disease or not. So these of course are very important immune molecules present on the antigen presenting cells. So whatever important genes I mentioned, you can see here, this is coded in chromosome 6. Now let me go ahead something. HLA, human leukocyte antigen, and how it's linked with the autoimmune diseases. If you see HLA-B27 is linked with the ankylosing spondylitis, reactive arthritis, acute anterior uveitis. Similarly, HLA-B47 is linked with that is if the population has these allele, they are more subject to get these kind of a risk of disease. 21 hydroxylase deficiency. If the population is having this gene, they are more prone to get these kind of a disease. Like this, you see, so many diseases linked with HL alleles are shown. So one day I'm telling you that nowadays in the developed countries, they ask you to come up not with a horoscope, but come with some kind like a genome patrika. The genome patrika has entire information about what HL alleles we have, and this can also in future we can able to predict what kind of diseases can happen. Of course, this is directed through genetic counseling. Okay, now let me go ahead with this next server other than IEDV. The Indian scientist, the famous scientist called Raghava, has developed so many databases for vaccine design and development. For vaccine design, one such T cell databases, he itself has himself has a creation of various tools for T helper epitopes, cytotoxic lymphocyte epitopes. It's great, isn't it? He belongs to I Square IT Delhi. Okay, and for T cell helper epitopes, we have MSCBN, ProBred, MHC, MHC2 spread. So we have plenty of tools, and the description of these MHC the tools are shown here. That is, most of the tools are distinguished with respect to machine learning algorithm. Each and every tool has a kind of a distinguished machine learning algorithm, and so the properties will be getting accordingly. And here in the cytotoxic lymphocytes, similarly, however, you find different machine learning algorithms are used for predicting helper T cells. You can also find similarly for this. If ProPred is used for helper T cell epitopes, ProPred 1 is used for cytotoxic T lymphocytes. Like this, you can find few more tools. The major URL to go access these tools is crdd. This is the URL. Of course, I pasted in the WhatsApp. Now, let me go ahead with the next thing. ProPred. Just now I told you, na, ProPred for helper T cell epitopes. So if I go to the ProPred, we will be getting this kind of a home page. We have to paste the sequence. What sequence we have to paste? So if you see my last session, do you remember I illustrated you something on Nipah virus protein W, because more all the structure other structural proteins and non-structural proteins already published in papers. So I tried with Nipah virus protein W. You remember now? So whatever W protein sequence from UniProt, first we have to download the FASTA format of a sequence from UniProt, but don't paste the entire FASTA, just paste the sequence alone. That is amino acid sequence alone you will be pasting here from the FASTA format. And then we can select the HLA alleles. After pasting the sequence, paste the select the HLA alleles. For example, if we want to come up with a kind of a vaccine to treat Kerala population, for the Kerala community, the major community of Kerala, Kerala Christians, so I can see what alleles are there for the Kerala or Keralites or for the Kerala Christians specifically, then I can select that alleles alone. And if I start submitting this sequence, we will be getting peptides, most appropriate peptides for that specific population. So this kind of approach, we are calling it as a precision-based vaccine design. Okay. So this is how we can try with any of the tools linked with the databases in immunoinformatics. Okay. Like however we have tried, we can also try the same even in IEDB. And this is something ProPred. So ProPred is related to MSc class 2. Okay, here still you have to paste the same amino acid sequence, that is Nipah virus W, and only the length alone you change. What kind of length we want. Okay, the properties will be in the bottom. You can change the length. Like if you want, this is MSc class 2. MSc class 2 refers to CD4. CD4 refers to helper T cell, isn't it? So this refers to the helper T cell. So helper T cell has a smaller peptide. Okay, that is 9. So here it is 9. Here in this case is 13. Okay. Okay. So am I right? Yes. This is related to the MHC2. Okay. So this is how we search the molecules. No, actually, I am mentioning something opposite. For HLA, ABC, we go with small. We go with a small size. With the HLA, DP, DQ, DR, we take the large size. So this is actually MHC2 large size, yes, large size. However, I mentioned that is if you see this MHC1 is related to what gene? MHC1 is related to CD8. CD8 corresponds to cytotoxic, and this leads to the CD4 helper T cell, and this should be a larger 13 amino acid. How come we can decide which is a larger, which is smaller? You can able to find from these genes. If you have HLA, DRP, TP, DQ, DR, then obviously we have to predict a larger epitope. If you have ABC, we have to predict a smaller epitope. Okay, now let me go ahead with the net MHC pan. If you see the net MHC pan, we have to paste still the same kind of the Nipah virus W protein in our case, however we tried. Similarly, we can paste the sequence. Here it can also accept the FASTA format. So, whatever I showed you that net MSC pan, this is from a different website. So IEDB is different, Raghava Observer is different, and this service is different. Like this, you have 100 types of websites to try with cytotoxic T lymphocyte epitope prediction as well as helper T cell epitope predictions. And in this, if you see, these are the parameters you can scroll. If you scroll down, you can find this. So obviously, whatever epitopes we predict, these are predicted based on the percentage rank. Okay, and this is the URL IEDB. So IEDB just now I told you no. So today we'll be discussing on T cell epitope prediction and epitope analysis tools. Now, how to predict that MHC1? So I told you initially we have to click this hyperlink. If you click this hyperlink, it directly takes you to this. Here we have to paste the amino acid sequence of any specific virus or cancer antigen, whatever you want to develop. If you want to develop a cancer vaccine, of course, cancer antigen will be pasting here. And then we have to select the size. So just because we are using the ABC, HLA ABC alleles, obviously I select defaultly 9 as the amino acid length of the epitopic peptide. And then whatever alleles we want to select, we can select here. But how to select the alleles? If I want to find for the correlates, how to make it? First, if only if I know the alleles, then I can select the alleles and whatever we had in the input as a default, we can predict the epitopes. Okay, to select how to select the alleles, now I'm going to take you to the browser. So for this, we have to go to the IEDB website. So we have to go to the IEDB website. Inside that, we can go to population coverage. If you go to the population coverage, which is present in epitope analysis tools, it takes you to this page. In this page, we have MHC1 separate, MHC2 separate, 1 and 2 combined. Our objective is to check the correlate people alleles. For this, allele frequency net database. You also have different countries here, but ignore that. I just click the allele frequency net database. You find here, isn't it? Now we have to go to the populations. Inside the populations, click browse populations by geographical regions. So after getting inside this URL, you can see this map, etc. So all this, whatever is shown here, this much population alleles are already, we can find the data for all these allele populations. Okay. Now let me go ahead with this. So these are HLA alleles and these are the specific regions. Where can we find India? So if you want to find Kerala, we have to find India. Can we go with South Asia? Are we in South Asia? Yes, we are in South Asia. In South Asia, can find Bangladesh and India. In India, 82 regions are specified, 82 communities. Okay, and specific regions are specified. Now, if I want to go to the Kerala, so Kerala, I think many remember Nair. Okay, can we go ahead with the Nair? So if you see the Nair, we have basically ABC, that is HLA molecules. HLA ABC, it means that MHC1 molecules information is present. But if I want to come up with even with the MHC2 molecules, can we go ahead with another major population of Kerala? So what might be the major population of the Kerala where we can find? So in India, Kerala, Malayalam speaking. So here other than ABC, we can also find the MHC2 molecules. TQ, is available. Okay, so the when compared to TP, you find these are the alleles. So better I can go here. Okay, India, Kerala, Malayalam speaking. So if you want to go with specific community, you can try it. Definitely, whatever you go with the epitope prediction, that is more precise for that community. Now I'm going to I've taken to the Malayalam speaking people of Kerala. Now click this allele frequencies. If you click the allele frequencies, you will see this much population, that is percentage of individuals that have the allele. Okay, that is A010101. You can see 16.3 percentage. This is the maximum, not this. We have one more, 31.2. A110101. Okay, so the maximum Keralites have this allele. So if you take these alleles, if you want to take top two or three alleles, you take this as the first allele, and if you want to take the second best allele is this. So one day you have to feed these alleles as an inputs there. Okay, so that you are targeting the maximum of Kerala population. You can also select everything if you want. Only thing is that you have to type. Okay. Now let me go ahead. So if I want to use this T cell epitope prediction, especially class 1, with our objective is to take an input. So the input what we are going to take now at present is Nipah virus W. To take a Nipah virus W protein, I'll be taking you to the fully annotated protein database called UniProt. Inside the UniProt, you can check for the Nipah virus W. Nipah virus W. So I'm giving the keywords. Let us say I'm getting that or not. Nipah virus L is there. How about W? If you know the Uniprot ID, we can type the Uniprot ID. So instead of simply searching a particular thing, if you know the ID, so I check it in my PPT whether I have that ID for Nipavirus W. So if you see this Nipavirus protein, I have P0C1C7. P0C1C7. We have to search for it. P0C1C7. If you patiently search, you will be getting, but the first page we don't get. So it is actually protein W of Nipah virus. Okay, our objective is to take the sequence of FASTA format. Take the sequence. Download 450 amino acid length. Download. So here we have to paste only the sequence. So ignore the first line. Copy the remaining lines. And whatever we have copied, we have to paste it. Defaultly, you see, you can able to see for the SARS spike glycoprotein. If you want to work any example, you can simply have to click this run button. But I want to try with Nipavirus W. So what I'm going to do, I'm going to paste my sequence. So whatever I pasted, okay, input sequence, we have to type the alleles. What are the alleles we have selected at present? The alleles we have selected is A110101. Okay. Uh, allele finder HLA, HLA, A01, I forgot, 110101, 11. So HLA, first, oh no, if I search it, it will be selected. HLA, or is it possible to copy and paste? Let me try. I copied. Now I'm going to try pasting the sequence, not working properly. HLA, A, 11. So I'm getting 01, isn't it? HLA 1101. So that is selected now. HLA 1101. There you have subtypes like 01. Other than this, 11, we also have 24. HLA 24. HLA 24. Okay. So 24 we can select the 24. If you want to select other 24 subtypes, you can select everything. So whatever we have selected, submit. So now the 9 is already in default, it is fixed. If you want to increase to 10, you can increase the 10. So all the things will be getting 10. And if you click one Here you get both the 9 amino acid peptide as well as 10 amino acid peptide. For example, if I click here, see 9 and 10 are shown. So whatever you predict, you have can see the epitopes with both the sizes. Okay. So now let me go ahead with this run button. You already pasted W protein sequence. We have kept two different peptide lengths, and we have selected two major alleles of Kerala population. Now I am predicting the run button. Similarly, we can try with other kind of community people also. Now let me see which is the see these are the results we are getting from the MHC1 epitopes. So whatever epitopes information you are getting, if you want to open it in Excel, click download. So all rows, TSV or CSV, anything can open. TSV stands for tabulated separated value file. CSV, comma separated value file. Both the files can be opened in Excel. So now this time I open in CSV because already TSV we tried with the string database. Okay, now I'm going to download the CSV format file in the desktop of AML DVD. Here, whatever I download is for the MSC1 peptide. Okay, MSC1 epitopic peptide. So I save it like MSC1 peptide.csv. Now, let me double click this Excel file and I just zoom in and show it to you. We have a peptide sequence here, and these are the lengths shown. And if you want to see which is the best kind of a peptide, from how many peptides, how many data are present. At present, 1529 peptides are predicted. In these two sizes, 9 and 10 amino acid length, you can see around 1529 epitopic peptides are predicted for Nipah virus W protein. But if you want to select which is the best one, so I just take you to the post. Here we have something called net MHC pan score. Okay, the higher the score is the best epitopic peptide. So I'm going to select this field, and after selecting this field, you will be going with the data filter. So in the data filter, we can check the largest value. The largest value, whatever we got, is 0.90. So that I select and click OK. So this is the largest value of the net MLC1 score. This is the peptide AVK EE PP QK. Okay, so this is the screened peptide of MHC1. Okay, now let me go ahead with the next MHC2 prediction. So till now, whatever I've shown is related to the MHC class 1. So here we can also predict the immunogenicity, processing, binding, elution, etc. So let me go ahead with this next thing, MHC class 2. So in MHC class 2, we can able to find the similar kind of a window. Only thing is that we have to paste the input of any viral antigen or cancer antigen or whatever related to autoimmune diseases. Now, if you want to fix a particular length like 13 amino acid, we can have 13. Okay, this is the 13 amino acid, and whatever we have, oh no, not 16. Okay, 15, or even we can drag and keep the 13 alone. If I want the 13 alone, I just change from 15 to 13. Okay, and now let me go ahead with the alleles. To find the correct alleles of for this, you have to go with that T, Q, or D, or that sequences. ABC alone is found. First, let me, because whatever we have selected, we have DP, DQ, DR, etc. So if you want to find, you have to check like that. Okay. Let me go to the next community where we have more of here itself they have shown, but it's not showing properly. So it means that the Kerala population has not matching of MSC, or even the data whatever is taken is not taken for this MHC2 genes. If you take this Madurai people, okay, Madurai people, we don't have even data for the Madurai people. So, for example, if you take this Uttar Pradesh people, we have MHC2 data. So, go to the allele frequencies. In the allele frequencies, we can able to see this information, and which is the major allele frequency in decimals. You see allele frequency in decimal. Uh, I am getting something like 0.1510 DQB1, DQB106. Okay, so DQB1 is the DQB1. Yes, DQA1 is there, but B1 is not there. Whatever I searched in the allele frequency, DQB1. So some information is also not available. If it is available, we can try, or else whatever we have DQA1 I selected, where you also find DQB1 along with it. So DQA1 and DQB1 is present at present. So whatever research we can predict run. So we'll be getting the peptide epitopes. Similar kind of a procedure we have to do. That is download the Excel file, and we have to, after downloading the Excel file, we have to screen the best epitope based on the score. So we have net MSC, two scores. So we have to see this net MSC2, and which is showing the best score is the best one. So data, filter. So this is the best score. So according to the top score, whatever we filtered, this is the best epitopic peptide of MHC2. So which is 13 amino acid length. So this is MHC2 based peptide, and whatever I predicted that is the MHC1 based epitopic peptide based on the scores from IEDB. Like IEDB, we have plenty of websites. So even in these websites, you can search and you can get this information. Now let me go ahead with the PPT where I stopped. Okay, in the presentation, I just mentioned you how to go with the T cell epitope prediction. Initially, we have to check with the epitope analysis tools. I told you how to go with the population coverage, find the alleles, and then you predict separately for MSC class 1 and MSC class 2. So like however we have done, pasting the antigen sequence, selecting the alleles, and go click run to find the epitopic peptides. So how to check the allele frequency? So for checking the allele frequency, go to the population coverage, click this allele frequency in a database. Then we have to go to the specific community people. Okay, and for working on this kind of a thing, you can use a reference article and you can check your results. So this paper I published in 2020 in combinatorial chemistry and high throughput screening for T cell epitope based vaccine design for Nipah virus. So there itself you can find the inputs, etc. You can, like however I've taken the Uniprot ID. that information is also available. You can take and you can work it, or you can try with the SARS-CoV-2 virus for any new strains, emerging strains. Now, let me go ahead with the next publication. The next publication is in a book chapter. Here also, I have mentioned so many things related to how to develop the drugs related to enzymes. I mean, enzyme inhibitors for different viral diseases. And this is the workflow. So, take any specific protein of a virus or a cancer, and then you start exploring the numerous epitopes. In our case, we got 1522. Out of 1522 epitopes for MHC1, we screened the best one. And similarly, we got 1000 epitopes for MHC2. Then we have screened the best epitopes. If you increase the number of alleles, for the first case, I used two alleles. But for MHC2, I used only single allele. If you increase many alleles, you get many epitopes. Then our objective is to screen based on the scores. Maximum the score is the best epitope. So Nipah virus genome, we have structural proteins like N, P, M, F, G, L. These are the structural proteins. A non-structural protein. This is a non-structural protein W. So already the scientists worked with all the structural proteins or non-structural. So I worked with this W of Nipah virus genome. Nipah virus genome is smaller in size when compared to SARS-CoV-2. The size is around 18.2 kb. And Nipah virus genome, how it looks like. Okay, this is mentioned. There is only one strain, there is no mutation. So, whatever we design, a kind of a peptide epitope, that is sufficient enough for treating the Nipah virus infection. But only thing is that for what alleles we search, that is more specific to the alleles. So T cell epitope prediction. In T cell epitope prediction, how this is predicted in the software? So for the it uses the features such as amphipathicity motifs that bind the MSC molecules. Okay, what makes the peptides bind with the surface of the MSC? Amphipathicity and motifs. Generally, machine learning techniques like AN and support vector machine, these are used for predicting this kind of binding of peptides on the surface of the MSC molecules. And of course, the prediction of cleavage and transport associated with antigen processing. You remember? So if you see that MSC molecules, that is especially the peptides are processed inside the cell, and finally, the MSC molecules are presented on the surface of APC. So during that processing, you can find TAP protein, transported associated protein. So the processing is dependent on this TAP. So this kind of a TAP processing can also be predicted in the form of score. TAP score can be predicted. So only thing is that if you have to paste the antigen sequence, then you get the TAP score also. Other than finding the best epitopes for T cell epitopes, we can also find TAP scores. Okay, these are intermediate scores. And the next thing is the T cell immunogenicity. For if you want to determine antigen processing, peptide binding to MSC molecules recognized by a cognate T cell receptor of T cells, we can use this T cell immunogenicity. So this can be predicted using this URL. In IITB, the immunogenicity, a separate tool is there. Simply we have to paste the antigen sequence to predict the immunogenicity. Conservancy analysis. The conservancy is a fraction of the protein sections that the epitope contains, and identity is a correspondence. Okay, so for predicting the conservancy, okay, we can use the URL like tools.immuneepitope.org. Okay, so through this, we can able to predict the conservancy. Next thing is toxicity. Whatever I'm telling is the physicochemical properties of the predicted epitope. So if you want to check the immunogenicity, if you want to predict the toxicity, we can use these kind of an URLs. Actually, this is not present in IETB. The toxin pred is a program. This is present in the Raghava web server. If you go to this URL and if you paste the kind of epitopes, whatever we predicted, it can predict us the toxicity. So earlier in drug design, we use the chemical molecules like drugs inhibitors. But here, whatever we try to get an output is peptides. So for this analyzing the properties of peptides, especially toxicity, we have to feed the peptide, epitopic peptides. So I suggest you, whatever peptides we generate in MSC1 and MSC2 prediction, don't simply restrict up to the best one. You predict up to best 10 or 20. So whatever you predict 10 or 20, you check the immunogenicity. And we also have to check the toxicity. Because even the fifth peptide or sixth peptide can give no toxic, but the first four peptides or five peptides can show the toxic. So try to get as many number of peptides, around to 10 to 20. Then we can analyze these properties. Then we can finalize which is the best epitopic peptide. Don't merely judge based on the scores we get from IEDB alone. You can also try with few servers like Raghava web server or other few servers. If you try with two or three or four servers, you can better judge which is the best epitope. Other than that, of course, you have to check with the toxicity analysis as well as immunogenicity analysis. Here in the toxicity analysis, this is predicted, okay, that is epitopic properties or predicted, the toxicity properties are predicted through hydrophobicity, hydropathic index, that is hydropathicness is what we call it as hydrophilicity, and molecular weight, net hydrogen, side bulk, and steric hindrance. So these are the properties used to determine the toxicity of the predicted epitopic peptide. So for this, first try with any specific antigen. So whatever I've taken is the uniprot ID, and you get this sequence. Whatever sequence I get, I just pasted into. You can give this as an input for T cell epitope prediction. Even for B cell epitope prediction, you can use the same input. And here, this is the strategy I used in my published paper. There is some mistakes here. So these mistakes I'm just uh, correcting now itself during my lecture session. If I want to predict a MSC1 epitopic peptides, I told you to go with 9 amino acid. And for the MSC2, I told you to go with the 13 amino acid. So whatever I'm telling 9 and 13, this is what prescribed or this was used by John Hopkins hospitals and other popular hospitals in USA. So the hospitals, okay, in their research, okay, in multiple publication, you can see these are the preferred sizes they kept. But you have in the form of a range, as I earlier mentioned, 8 to 11 and around 13 to 17, like this. Okay, now let me go ahead with this. Instead of trying with these amino acid, I just use the 9 amino acid. So that is wrong, but you can correct it. Okay, it's already published, but hereafter, if you want to try with these kind of a sizes. So if you see this predicted T cell epitopes, okay, I tried with the different servers, server 1, server 2, server 3, server 4. So I predicted from four different URL servers, and whatever I'm getting the scores, I just showing it here. So whatever I'm getting, I can't simply tell that this is the best MHC1 peptide, this is not a best MHC2 peptide. Other than getting the scores, the next step of what I done is predicted the immunogenicity, conservancy, and toxicity. So whatever peptides I predicted, I use the same peptides to check which is the best one. So when I start predicting it, so I can able to see that the higher the immunogenicity, I screened as a best epitope. So out of all these epitopes, this epitopic peptide of MHC1 has shown a good score. Not only that, it shown non-toxic. Okay, so I finalize this is the best epitopic peptide of MHC1, that is HLA. And then I gone with the MHC2. In MHC2, I just found the two best epitopes. So from that, which is best, I screen based once again to the immunogenicity. The higher the value is the best kind of immunogenic molecule, and it's of course non-toxic. So this is the next MHC2 molecule. Okay, so these are the predicted epitopic peptides. After this, I've gone with molecular docking. So I dock this peptide with the protein through peptide protein docking servers. So for what protein I docked? The protein I docked is the human allele. For example, in the Kerala population, we have taken HLA. What is that? HLA A24, HLA A11. So whatever alleles I've taken, okay, we can take that protein sequence even from NCBI. I've used the NCBI GenPep database, and I searched for a particular allele, and whatever allele protein sequence I got, I predicted the structure using Swiss modeler. As such, you won't get a structure for any specific allele. We have to find the protein sequence and we have to predict the protein. After predicting the protein structure, I just docked the peptide with that. So this is for MHC2 peptide. And this is for MHC1. For MHC1, whatever we screened, this is the best screen. And this is for MHC2. I showed you just now in the previous slide. So for MHC1, this is the peptide. So this peptide I just docked with this allele, specific allele. Okay, whatever is more prominent to a particular community, so that allele protein sequence I take and predict a structure and try to dock with the best screened epitope. And this is what I showed as a scores. Okay, here we are not concerning on the significant scores. Like actually, I told you in Autodoc, we go with the binding energy. If it is less than minus 6 kilocalorie per mole, I told you that is a significant one. This applies only for the small molecules. Peptide is a medium molecule. So this won't apply for this. So whatever score you can get, you can show the scores. Okay. And the next what I've done is the molecular simulation. In the molecular simulation, whatever peptides I founded with that human proteins, I just tried with RMSD. So under this case, you see, it shows a stable thing. Okay, that is, it shows around 0.75 RMSD. Okay, from here to here, that is around 30 to 147, something like that. You can see the RMSD is stable and it comes around 0.75 Newton meter. Okay, so no, not Newton meter, nanometer. RMSD 0.75 nm, and these are the time. So up to 30 to 147 nanoseconds, the peptide is binding stably. The entire peptide is binding stably with the entire protein. Okay, in this particular time. In RMSF, it tells about the flexibility or with respect to the amino acid residues. Whatever amino acid residues we have, we can see 0 to 200. We find the maximum kind of a peak, which comes around 0.55. I told you if only if in RMSF we get more than 1, our peptide is not binding stably. It is having some kind of unstable interactions. Just because we have 0.55, which is less than 1, we can still can tell all the amino acid residues also has a stable kind of interaction with the peptide. And now let me go ahead with the area. We have types of area: SA, MMPB, SA, LIE. So many types of area is there. If you see the area, how the surface area of the peptide is binding with the HLA molecules. If you see this, this shows some kind of a constant area from this time to this time. So you can show that, and what is the area, that alone you can mention. Okay, the peptide surface area covering current binding with the HLA molecules. And radius of gyration tells about the moment of inertia. So here also you check the stable thing. Okay, so generally MD simulation is used to check the stability of binding of peptide with proteins. Okay, over a period of time. In all these cases, I have used a simulation time of 200 nanoseconds. For publication, nowadays they're asking up to 200 nanoseconds. Okay, so even if you can see many publications with 500 nanoseconds or more than 500 nanoseconds also. Okay, so based on the publication quality, we can able to extend our simulation time. So okay, this is the thing already uh, we have discussed, and that is what are the popular adjuvants we are working in our lab, and this is the tool we have developed to for the plant-based adjuvants for any diseases. For autoimmune disease, for cancer disease, we can check which is the best plant compound can be used as an adjuvant, okay, for the upcoming vaccines. And these are the plant-based adjuvants I tried in my research, Pyrivinon B and Withnoid A. So first we have to, if you want to check whether these adjuvants work effectively or not, we have to procure the vaccines without adjuvants, and then we have to try with our adjuvants. So we also have vaccines with adjuvants in default, but if you have without adjuvants, we can try without adjuvants. Then only we can distinguish the uh, beneficial effect of the adjuvants. Okay, so this we have tried with both cellular responses as well as humoral responses. Adjuvants in COVID-19 vaccines. So we have so many vaccines, isn't it? So in Covishield, which is used as an adjuvant, adenovirus coding spike protein. Adenovirus is used as an adjuvant. Of course, there is a component, and this component is also act like an adjuvant in Covishield. In Sputnik, again adenoviral vector. This is used as an adjuvant. Covax, what they have used? Saponins, plant-based saponins they used as an adjuvant. 40 nanometer nanoparticles of saponins along with the cholesterol and phospholipid. So together, all these three, okay, we are using as an adjuvant for Covax vaccine developed by Bharat Biotech. Like this, you can see plenty of adjuvants examples. And if you see the functional diversity of the adjuvants, we can see the most widely used adjuvant, the inorganic adjuvant used in most of the vaccine is the alum. Okay, this is used for sustained delivery. But nowadays, to avoid the accumulation of inorganic metals, we are using the plant-based adjuvants. So, synthetic adjuvant like phytol-derived immunostimulant, phytonol, is also used to augment the immunogenicity specific IgG1 and IgG2 antibody responses. What are the future projects you can work? So that in 2022, you can find this second wave mutations in 21 to 22. At the time, in the 1330 amino acid, 1300, I believe the spike glycoprotein size is around 1300 something. So whatever we have, in the particular amino acid position, if lysine is mutated to aspargine or 309, okay, this called as a second strain. I mean, this is another strain of SARS-CoV-2. So for this, we can come up with a new vaccine other than Covishield and Covaxin. Similarly, if instead of leucine, if it is mutated as arginine in 452 position, this is another strain. This is one strain of SARS-CoV-2, this is another strain. Like this, we have plenty of strains, hundreds of strains of that. For each and every strain also, we can come up with a vaccine specifically to the population. So I have completed this session on T cell epitope prediction, and tomorrow we'll be discussing on B cell epitope prediction. So any doubts, you are welcome to ask now. Any doubts? If no doubts, thank you very much for your patience and interaction. Have a great day to you all.